derivative come from the natural product. For example, you know codeine, you know morphine, you know toxotexan, tapotexan, and then vincristin, vinbrancin, and so on. I think this is come from the natural product. This is very interesting, the seminar, because no, many other researchers is going to the simple screening in silico. Because screening in silico, I think, is very cheaper, very fast, and very get many other database to going to implementation in vivo, and then we also look about the toxicity, and then we are going to clinical trial research. I think it's the better we have the big data come from the in silico research. And then translational pharmacology did, I think, the basic research to clinical application. Yeah, because I have many other clinical trial come from the basic how to look about the in vitro activity in vivo. But I think the in silico is new screening drug paradigm. Next, research area. I think we have to discover of the active compound come from the natural product resource, especially in the traditional herbal medicine. Yeah, how to determine to the molecular mechanism. For example, one is interesting network pharmacology. Yeah, why we must look the network pharmacology? Because I think the natural product, I think, have many multi-compound ingredients. Yes, I think in silico validation, I think in vitro validation, because I think in silico, this is the first screening activity. I think we have looked about the possibility when we in silico positive and possible in vitro and in vivo and clinical trial is have the activity. Yes, the goal of the research area, I think, how to develop the new drug candidate for non-communicable disease. Yeah, I have joined together with the Prof. Royha activity, research activity, how to discover about the candidate of the anti-cancer activity. Next. This is network pharmacology research area. Next again, yeah. Next slide. Yes. Next, networking pharmacology. Next. Yes. Yeah. This is the big problem. How to develop the drug discovery come from the natural product, yeah, because the herbal compound is very multi-compound ingredient, different with the synthetic drug. Synthetic drug only one compound active, but I think the implementation on the network pharmacology is good prediction mechanism, yes. And then we get many other database and visualization, and we can use to implementation when you are develop to in vitro activity, in vivo, and in clinical trial. Next, this is the network pharmacology. Yes, the concept how the network pharmacology is interesting to implementation to drug discovery. Because herbal medicine is different of the single compound. Because the herbal medicine, I think, have the multi-compound and the activity possibility is synergism effect. Yeah, synergism effect. For example, my student PhD program is isolation alkaloid indole come from the Mitragina speciosa. The name is kratom. I think when we isolated single compound mitraginin is lower activity of anti-cancer activity in breast cancer cell line. Yeah, I think this phenomena, but the activity, I think the multifactorial effect is possibility. We look about the network pharmacology because one active compound maybe is have the only one target, but possibility one compound is have more than multiple target. Next, this is the concept is networking pharmacology. Yes, medical plant is have many other multiple compound, and when we have one compound, I think have one target in marker disease. For example, in anti-cancer activity, we have used marker compound, for example, anti-apoptosis protein, BCL2 possibility, and we have also, we can look about the caspase-3, caspase-3 possibility to look good target in develop of the anti-cancer. Because one disease have possibility 1 and 2, 3 is biomarker. Next, this is state of the network of pharmacology, how to look about the metabolic compound come from the extract possibility. I think it's very simple analysis today because the liquid chromatography tandem with mass spectra, I think it's very easy to look about the multi-compound in the ingredient in the extract. And then we are going to look about the gen in the database. And then we are to also implementation to interaction by the network pharmacology. And then we are also to get the potential metabolic compound active. We have the networking network pharmacology. We are also to develop to in vitro and in vivo and clinical trial test possibility. This is next compound database. I think we have also easy to get in many other database in the internet possibility in USDA, the phytochemical, and then I think it's very simple in the last activity, liquid chromatography as tandem mass spectra. Yeah, in my laboratory in Erlangga University, I think it's until now have the new liquid chromatography tandem mass spectra. I think it's very better when the many other students, lecture, researcher, in the other participant to look about the use the liquid chromatography tandem mass spectra in Erlangga University until now is ready to get sample come from many other researcher. Next, this is target gen database. I think next one slide again. I think today is very easy to get the information about the disease target. Get database, we can you are can to download, we can to uses this GenNet. I think the very very much information about the disease in can to use the target molecule when you implementation of network pharmacology. Next, this is also how to determination of overlapping compound come from the ingredient in the extract multi-compound. I think it's not difficult because we get the database come from the database DigiNet, and then also we get LCMS mass spectra tandem. Is you can look about the many other compound come from the extract the plants in Indonesia, and then we can implementation, and then we can look about the overlapping, yeah, many other gen target we can use to look about the interaction of networking with the metabolic compound. Next, This is compound target. Next, yeah. For example, compound target with the gen database we can use. I think I don't know. This is the berbayar atau tidak database ini? Yes, no. I think the free, yeah, database target gen database we can use free. Student and the lecture and the participant seminar we can use the target gen database. Next, this is visualization network pharmacology. For example, this is research come from Professor Royha and team, and I also the member in the research about the how look networking pharmacology come from the plant in Indonesia name is the kayu kuning, yeah, kayu kuning. This is many other compound come from the kayu kuning is have identification and then clustering. This is many other compound is flavonoid, coumarin, and so on. This is how to look about the many other molecule target, many other proteins have the interaction with the compound come from the family of Berberidaceae. Berberidaceae or yeah. Okay, we can look the next slide. The protein-protein interaction. I think there's very complete interaction. We can look about the one compound more than possibility interaction with the more molecule target. Yeah, for example, in hypothesis, in paradigma about the interaction networking. I think the multiple compound is also one. Compound activities have more than one protein interaction in pharmacology implementation networking. This is how to look to continue about the interaction of pharmacology. I think many other graphic, many other information can you get next? Yes, I think go and pathway analysis. I think many other program to get to analysis about the networking pharmacology. Yeah, next. Next, yeah, this is the profiling Archangelesia flava root, yes, the akar kayu kuning. How to exploring therapeutic potential. One of the compound active is very have the high potential activity in when we look about the networking pharmacology. The name is the oxoberberin compound. I think oxoberberin compound is the compound of the alkaloid compound. Is very high concentration in Archangelesia flava. Yes, Archangelesia flava. I think the endemis in the West Indonesia, for example, in Northwest, tapi East Indonesia, for example, is Nusa Tenggara Timur and Maluku and so on, and also in Borneo Island is very endemis in the Archangelesia flava. But I think the Archangelesia flava is many other activity because my group research in Erlangga University also to look about the osteoarthritis activity. Yeah, until now the Professor Retno Widyawati is to look about the anti-inflammation activity, and then we are also to develop to topical product to look about the activity of the arthritis rheumatoid. Yes, I think the have the possibility connect about the anti-inflammation and also anti-cancer activity also. Yes, this is the metabolite profiling is have done, and then do pharmacology networking analysis and in silico and in vitro evaluation also. But I think many other research possibility next, usually not a correlation about the in silico and networking pharmacology and in vitro and in vivo activities usually is not linear activity. Possibility in the in silico activity is very good interaction with many other protein possibility and many other marker target in the cancer activity. Yes, I think it's better when the finish in the in vitro activity, in silico activity, we must going to in vitro activity and then continue to the in vivo activity. But Archangelasia flava, all of participants, I have information for you because in the Federation Food Drug Administration in Indonesia, Badan POM, Archangelasia flava is I think the forbidden to develop traditional medicine because introduction the Archangelasia flava is have the berberin alkaloid compound possibility have the side effect in the many other tissue in human. I think the better when many other researchers, for example, Prof. Royha and team, to look about the toxicity about the extract come from the Archangelasia flava. Yes, I think it's the better when we look for the toxicity. Next, this is chromatogram of Archangelasia flava with use the liquid chromatography tandem mass spectra. This is the mass spectra liquid chromatography. Possibility is very difficult to identify the one compound because one peak possibility more than one compound is also have the similarity in one peak. Yes, one peak possibility more than 4, 5 compound is also have the mass spectra is have the identified is SIM possibility. Yeah, I think Prof. Royha is also to look about the one peak possibility more than one compound is have one peak in the retention time in the liquid chromatography tandem mass spectra. In my institution, Erlangga University have a new liquid mass spectra and tandem mass spectra is have the library ingredient in the LCM SMS. Yes, this is I think the very to helping you to identify one peak because one peak possibility not one compound also. Next, this is the identified of the compound. I think the oxoberberin is high activity when to look about the networking pharmacology. Next. This is also the prediction of the Archangelasia flava come from the root extract about the identified about the in silico activity. Yes. Next, this is a diagram network topology compound from Archangelasia flava. We can look about the many other compound, for example, flavonoid, coumarin, alkaloid, and this is as many other molecule target to get interaction. Yes, I think possible to look how to interaction is very complex. Yes, but we can look about the one compound come from the multi-compound come from the Archangelasia flava is can identify possibility interaction about the molecule target in the cancer, breast cancer. Sorry, yes, in breast cancer modeling. Next, this is also protein-protein interaction about the compound in Archangelasia flava. I think the better when we are look about the mechanism of the anti-cancer, we can look how to the compound active to induce of apoptosis, induce to the many other activated from the protein suppressor gen, for example. Yes, we can look about the many other signaling is the mechanism of angiogenesis protein, for example, is vascular endothelial growth factor, for example. Yes, one is the apoptosis induction. I think the suppressor gen induction also apoptosis, and then we also look about the metastase process in angiogenesis. We can use the marker protein. I think one is very good marker protein is vascular endothelial growth factor. Next, this is also ontology gen in pathway analysis. Yes, many other implementation of analysis of pharmacology networking we can have done in this process. Next, this is interaction about the compound active from the Archangelasia flava with many other protein in the cancer, breast cancer gen, breast cancer protein, breast cancer of marker cancer of marker tumor. Yes. Next, this is the docking molecule come from the high possibility activity oxoberberin. Yeah, oxoberberin. I think the compound active is very high concentration in the root of the Archangelasia flava. Yes, this is many other possibility interaction with the BCL2, CDK1, VEGF, so on, and many other protein peptide it can use to the interaction about the molecule docking. Next, this is also to look about the how the induced apoptosis from Archangelasia flava, especially is oxoberberin. Yeah, we can look about the induced by in vitro activity apoptosis. We can use the flow cytometer. Yes, the flow cytometry process implementation. You can look about the how many early apoptosis and late apoptosis we can implementation in the apoptosis in flow cytometry. Next, I think the last next one again. Next, yes, yeah, okay. I think this is not compound, not anti-cancer, but I think I have the project also with the Prof. Royha. We also to look about the systematic review about the aphrodisiac potential traditional medicine in Indonesia. Yeah, but implementation about the networking pharmacology when we are going to screening many other review article. We have many other article to look about the implementation about the networking pharmacology. Next, we have one candidate. I think Pasak Bumi. Yes, Pasak Bumi is Aerochroma longifolia. I think it's very strong identification about the networking pharmacology in many other protein come from the aphrodisiac activity. For example, many other protein, many other receptor, many other gene is interaction with the many other compound active come from the Pasak Bumi is ingredient is very popular in Borneo Island, yeah, in Kalimantan. I think the Pasak Bumi is very traditional medicine is implementation to the aphrodisiac activity. Yes. Next. Yes. This is meta-analysis, and then many other. Next. Yeah, traditional use in the part uses, doses, and the compound in traditional medicine to uses pasak bumi. Next. Yes. Yes. Next, I think many other empiric data, yeah, how to traditional use, part of uses and administration, yeah, many other administration use, I think the oral method, decocta, and so on, powder also, and then the many other activity, possibility in to induce by many other compound, for example, terpenoid, yes, phenolic and flavonoid. Next, this is diagram and come from the metabolic compound active from the pasak bumi, Ericoma species. How to look upon the protein is the dysfunction erectile dysfunction, erectile dysfunction. I think many other protein is more than yeah, many other proteins have the interaction with compound active. For example, stigmasterol, erocomonan, and ericomalactone. Yes. Next. This is compound active come from the Ericoma longivola. Yes. So we have many interaction with the gene target and the protein target. Next. Yes. I think seem like the topology of networking pharmacology in the area Ericoma longifolia with the first information about the anti-cancer interaction. Yeah, we can have many other profiling, many other picture, many other pathway. We can look about the interaction about the compound active if have the inhibition about the erectile dysfunction in the aphrodisiac effect. Yes. All participants, I think I have many short information about you about how the networking pharmacology is very interesting to look about the early screening. Yeah, because the networking pharmacology is the in silico screening. But in silico screening is very good to look about the possibility new plant it get to develop to many other activity, for example in anti-cancer activity. Yes, I think this is my presentation is a short result about the how implementation networking pharmacology to look about the activity in the disease, communicable disease, yeah, non-communicable disease, for example, anti-cancer, anti-diabetic also, maybe possibility in the Alzheimer disease is very interesting to look many other possibility come from many other component in Indonesia, and then also diabetic, Alzheimer, anti-osteoarthritis is also a good to one cluster is non-communication disease in groups and the anti-inflammatory, because inflammatory also to induce the anti-cancer and the metastasis and going to angiogenesis processes. Yes. I think thank you for your attention. This is the short information about the my research and group collaboration, and I think it's very better when the after finish the seminar international we can to join collaboration to many other researchers. For example, the one presenter come from the Thailand, and then we also come from the Korea, possibly, yes, Korea, the three. Malaysia, also Malaysia and UIN, and then we also join together with my group research in Erlangga University. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much for Prof. Sukardiman for the truly enlightening presentation of natural product-driven drug discovery and translational pharmacology for non-communicable disease. Okay, let's continue with the second speaker, which will be presented by Associate Professor Dr. Thiravan Laumetachit, a leading expert in computational biology, mathematical biology, a leading biomedical system from King Mongkut University of Technology, Thonburi, KMUTT, Thailand.
Okay, thank you very much. Thank you again for inviting me to present my research today.
Sorry, Dr. Thiravan.
Yes, can you hear me?
Is the connection still have trouble or is it safe?
Yeah, I think I can hear you very well. Can you hear me? Can you hear my audio?
Okay.
Yes, can you hear me?
Yes, I can hear you.
Okay, all right. Okay, thank you very much.
Yeah.
So,
okay, Mr. Thiravan, the floor and the screen are now yours for the next 30 minutes. For Dr. Thiravan, the time is yours.
all right. Thank you. So my topic today is applying computational modeling and AI to elucidate gene-protein regulatory networks and identify therapeutic targets in diseases. So the objectives of my research is to understand the complexity of the interactions between gene and proteins that regulate cellular functions and cellular responses to the environment. For example, in this picture, it shows two pathways, the MAP kinase pathway on the left and the PI3K AKT pathway on the right. And there is crosstalk between these two pathways. And it is interesting because if we apply, say, CI 1040, which is a drug that inhibits the MEK protein to suppress the proliferation of the cancer cells, it turns out that the cells, especially the cancer cell, can respond to this perturbation by switching on the right pathway and remains proliferative. Now the cells is resistant to the drug. So that's why we can combine CI 1040 with another drug, which is PIK 90, which is an inhibitor of PI3K. And by combining these two drugs, we can effectively suppress the proliferation of the cancer cells. So this is called drug combination when we apply two or more drugs to achieve a total effect greater than the sum of individual drugs. So for example, in this experiment, when we treat this breast cancer cell line with individual drug alone, for example, we can use the MEK inhibitor or a PI3K inhibitor. They can lower the viability of the cancer cells to certain levels. But then when we combine these two drugs together and use only half of the original dose, you see that the combined effects have a very effective outcomes in lowering the viability of the cancer cells. We can also even lower the doses of drugs to only 2.5 micromolar, and still we still see that the viability of the cells is really better than when we compare to the individual drugs alone. So that's the problem that I really want to understand. If we can understand the functions of this pathway, how they function together, interact together, then maybe we can understand and identify new therapeutic targets and also design more effective intervention strategies. And our lab approaches this problem using 3 methods: mathematical modeling, machine learning, and network analysis. So today I would like to share some of our research that use these techniques. So the first technique is mathematical modeling. So in this technique, basically we try to translate the biology of the interactions between protein and genes inside the cancer cells to a mathematical representation using equations or algorithms. And once we have this model that represents our system, we can use this model to simulate the behavior of the cells, how the cells respond to different treatments. We can also use the model to predict new scenarios, new treatments in our computer before validate our prediction in the experiments. So we use a specific approach called Boolean modeling. So in this modeling technique, we represent the activity of each component in our network by 0 or 1. So 0 means inactive and 1 means active. So for example, here we have a tiny network, the interaction between 4 proteins. We have protein X and Y that promote the activities of protein A. We have protein C that inhibits the activity of protein A. We can translate this interaction into this equation. So in this equation, we say that the activity of protein A in the future time step depends on the current activities of protein X, Y, and Z. So here X and Y are the activator, and C is the inhibitor. So by compute the interaction between these proteins of the input and compute the activity of the protein output, we can map the activities at the current state to the activity of the protein A, which is the output in the future time step. So we use this idea to simulate the change, the dynamics of protein activities inside the cancer cells. And we also apply a very specific technique called stochastic modeling. We add some probabilistic terms into our equation so that every time that we simulate our model, we have a slightly different outcome. And we use that to simulate many cells. So every time that we repeat our simulation, it will represent 1 cell. For example, here we have 3 cells that would have a little bit different behavior of the protein activities over time. Now what we do is that we simulate our model many times, like to simulate 1,000 of cells. And then we average the activities of protein to represent the population level protein activities. And now you see that we have the protein activity profiles that go up and down continuously. And that is the result that we can compare directly to experiments. So we use this idea to construct a very big model. So this model has 117 protein components, and we collect the interaction between these components from 3 databases. And my PhD student carefully put them one by one together and translate all these interactions into the equations like the one that I showed you in earlier slides. So for example, we have IL6, which depends on the activity of I61B or IL6. for example. So we have 117 equations, and then we update the activity of protein one by one. Okay, so let me show you some example from our model simulation. So this is a tiny part of the bigger model that we have to just illustrate what we can get from simulating the model. In this network, we have PI3K that activates AKT, mTORC1, X6K1, and this X6K1 inhibits another protein IRS1. So what happened is that when we try to inhibit this node or this protein using a PI3K inhibitor, so this represented by the simulation outcome here. So we apply the drug, the PI3K inhibitor at this time point. And you can see that in our simulation, PI3K activity turns off immediately, right, when we apply the drug. But what we found is that when you turn off the activity of this protein, AKT is off, right? mTORC1 is off, S6K1 is off because they are the downstream of the PI3K protein. But because S6K1 is the inhibitor of IRS1, so when you turn off the activity of this protein, then the activity of IRS1 goes up, right? Because the inhibitor of this node is gone. And you can observe that effect in our simulation result. You see that when we apply this drug, PI3K is off, but then IRS1 and the downstream of this protein, which is JAK2 and STAT5, go up. And we interpret this as the cancer cells switching from the PI3K AKT pathway, which is turned down by the drug, and use another pathway to remain proliferative. Okay, so our simulation can predict that if you combine this drug, PI3K inhibitor, with another drug that inhibit JAK2 or STAT5, that would have very effective combined effects on the proliferation of the cancer cells. And that is confirmed by another experiment. Here we see that when you apply the combination of these two drugs together, you can reduce the mass of the tumor cells to this level compared when you apply individual drugs alone, like this. So we use this framework to study 42 synergistic drug pairs. So we know that these 42 drug pairs are really effective in killing breast cancer cells. And we want to understand what are the mechanisms underlying the synergistic effects between these drugs. And we simulate our model and then cluster these 42 drug pairs into 5 patterns. So our study showed that if you can collectively promote the activities of FADD and BID, that will synergistically promote cell death. You can also collectively downregulate BCL2 to turn off the apoptosis and also promotes cancer cell death. You can also collectively inactivate mTORC1, ESR1 or cyclin D, and this will all have a very good effects in killing breast cancer cells. For the second project, so in the first project, we know that our simulation works very well in capturing the behavior of the cells. But because we have 117 proteins, it's quite difficult to identify which proteins that are important in driving the proliferation or having the effects on the effective treatment of the drugs. So in this second project, we try to use the machine learning approach to identify or pinpoint the key genes or protein responsible for effective drug combination. So the idea is that we have a data set of drug combination experiments from the DRUGcome database. So in this database, each experiment has the detail of the drug, the first drug, the second drug, the cell line that it was treated, and then the synergistic score. So this synergistic score can have negative and positive values. They are highly positive number if the two drugs here, A and B, are highly synergistic to each other, meaning that they work together effectively in suppressing cancer cell growth. Okay, if the number are highly negative, it means that they are bad pairs. They don't work well together. Okay, so what we did is that we have this data set from the experiments. We have our model from the previous project, right? So we take each pair in the database and simulate our model to get the activities of the protein, 117 proteins from our simulation. And we use the activities of this protein, predicted activities of the proteins, to use them as the input to build a machine learning model called random forest to predict the synergy score. So basically, this algorithm will select important proteins to put in the algorithm and use them as the decision making criteria to predict the synergistic score here. Once we have the model, we can use the model to predict the synergistic score as well as the contribution from each protein. So for example, if I input the combination of these two drugs, ACD5360 and fulvestrant into my model, it will predict a synergistic score between these two drugs. And it also prioritize or rank the contribution of the key proteins that contribute to the synergistic effect of these two drugs. For example, this particular example, we identify that there are 3 proteins which are FOXO3, NF-kappa B, and mTOR-C1 that are the key drivers of the effectiveness of this drug pair. And based on that, we can use the proteins that the machine learning selected for me to understand the underlying mechanisms between the combination of these two drugs. So it turned out that when we try to inhibit AKT, right? So we have the first drug is ACD5363. This drug inhibits the activity of the AKT protein. And AKT is the upstream regulator of these 3 key driver proteins. AKT activates NF-kappa B and mTOR-C1. So by suppressing the activity of this protein, we turn off NF-kappa B and the proliferation. And also we turn off mTOR-C1 and the protein synthesis. So this will suppress the proliferation and the growth of the cancer cells. But very importantly, that AKT also inhibits FOXO3. So when you have this drug, you try to shut down this left 2 pathway, right? But you turn on the activity of FOXO3, and FOXO3 activates estrogen receptor 1, which promote another pathway that make the cancer cell become proliferative. Okay, so that's how the cancer cell become resistant to the application of this drug. So our model predict that if you combine this drug and another drug, fulvestrant, to turn off these 3 pathways simultaneously, they will be very effective in killing breast cancer cells. Okay, so that's how we use the combination of mathematical modeling and machine learning approaches to understand the mechanism underlying the interaction between these proteins and come up with new or potential therapeutic targets. The last approach is network analysis. The model that we have in the previous 2 studies, they are a big model, right? 117 proteins. But still, we know its limitation that the actual networks that operate in the cancer cells are much bigger than that, right? They must contain thousands of genes and proteins. So we try to expand our network to include more components. So for example, in this case, we have a network of about 2,000 genes or proteins and about 160,000 interactions between them, which is difficult to do mathematical simulation. We cannot manually generate equations for each of these interactions, right? That's too much even for a PhD student, right? So we came up with another approach, which is network analysis. And the idea is that if we apply this drug, MK2206, we know that the target protein of this drug is AKT1, 2, and 3, right? So we know that if we apply this drug to the cancer cells, these 3 proteins will be downregulated, right? The activities will be downregulated. But we want to know that because these 3 proteins also are interconnected with other proteins, right? So we want to see how the effects, the perturbation of these 3 proteins can be propagated to other nodes in the network based on the interaction between them, right? And that is what that is the technique called network propagation. Here is an example. For example, if we have this network, right? If we know that the interaction between these 6 or 7 proteins in our network, and we know that if we use the combination of drug A and B, that will downregulate the activities of protein C and D, then we can propagate the perturbation of these 2 nodes to the rest of the node in the network using this kind of equation, which is the network propagation technique. So if we start from this vector, right, we have 1 and 1 indicating that our combination have a perturbation effect on these 2 nodes C and D. After we propagate the perturbation on these 2 nodes into the network, we will see that there are varying degree of perturbation on different nodes in the network, which can be represented by the color here. Okay, so you see that these 4 proteins will be affected by the combination of these 2 drugs more than the rest 3 nodes in the network. And this idea, we can apply this very quickly to all the drug pairs that we have in the database, right? For example, we can have drug pair 1 that targets protein A and B, drug pair 2 that targets protein C and D, and drug pair 3 that targets protein F and G. So you see that these 3 drug combinations, they have completely different targets, right? But after we do the network propagation, we can see that the first 2 drug pairs or first 2 drug combinations actually have similar effects when we consider the whole network, right? When we propagate the perturbation from node A and B versus the propagation from node C and D, they have similar profiles, right? Compared to the combination drug pair 3 that they have the perturbation effect at different corner of the network. Okay, so based on that, we can use these propagated profiles that we get to do clustering analysis to group them together, right? Let me remove the annotation. Okay, so we can do the clustering analysis and group the combinations into different clusters. For example, in this case, the combination 1 and 2 will be grouped together in one group, and drug pair 3 will be in a separate cluster. Okay, so you see that starting from completely different target of the drugs, right? Now we can converge the effects of this combination and see which combination that would have similar effects to the networks and the cellular function of the cancer cells. So what we did is that we analyzed 607 drug combinations tested on the 549 lung cancer cell line. And then we identified that there are 92 highly synergistic drug combination. These are the drug combinations that are very effective in treating these cancer cell line. And it is really interesting because we found that the 92 drugs can be grouped together into a big single cluster. And these 92 drugs have one single characteristic, which is the perturbation effect is very high in what we call metagene 2, right? You see this pattern, right? Okay, so metagene 2 is a group of gene or proteins in our network that collectively regulate certain specific pathways, and I will show you next what they are, right? But this is what we found that when you analyze different drug combinations that have different targets protein, they converge into similar pattern, right? They have a common mechanism that would responsible for the effectiveness of the treatment to this lung cancer cell line. We also confirm that if we compare certain drug combinations without the signature or the fingerprint of metagene 2 compared with another group that have the fingerprint or the high activity in metagene 2, they have significantly different synergistic effects. Okay, so the first group, so this is dasatinib combined with a group of drug here. And this group, this cluster, doesn't show high activity in metagene 2. And the second group is also dasatinib combined with another set of drugs. And this group has a very high activity in metagene 2. The right-hand side, I compare the distribution of the synergistic score of group 1 and group 2. And I show that group 2 with high metagene 2 activity have a significantly higher synergistic effect than group 1. So this confirms that by co-targeting pathway that is responsible for metagene 2, we can have a effective treatment on the lung cancer cell line. Okay, so finally, I look at the genes or the proteins that are the most contributors in metagene 2, and then you did the cake enrichment analysis, and the result is that top gene in metagene 2 are enriched in these two pathway, PI3K AKT pathway, and the last MAP kinase pathway. So our study suggests that if you can combine drugs that co-target these two pathways together, right, the PI3K AKT pathway and the last MAP kinase pathway together, then it would have a potential to suppress the proliferation of this particular lung cancer cell line very effectively. And so for the conclusion, we apply different computational techniques to study the pathway inside the cancer cells, and each approach reveals different underlying mechanisms, and that from those knowledge we can design or we can identify new therapeutic targets and also design or develop new drugs that would be more effective than the one that we have. For example, we use mathematical modeling to successfully study the dynamic crosstalk between pathways, and we identify combined therapeutic targets such as the inhibitors of PI3K and JAK2 that would be very effective in treating the triple negative breast cancer cell line. We use machine learning to identify key proteins such as mTORC1, NF-kappa B, and FOXO3. That is very important proteins in estrogen receptor positive breast cancer. And lastly, we also identify that if we can co-target, co-inhibit the PI3K AKT pathway together with the MAPK pathway, that would be a common strategy to overcome the proliferation of the lung cancer cell line. Okay, and for future direction, we did all the work in computers, right? It's just in silico study. So we would like to very happy to see if we can validate some of our prediction in the experiments. Right now, the direction of my research is also trying to apply these frameworks that we develop to integrate individual patient profiles like genetic mutations or transcriptomics data from subject-specific patient, right? And try to see if our framework can be used for personalized treatment design for individual patients. So I would like to thank my team, especially my student, Dr. Kitisak Thauma, my former PhD student, who worked on the breast cancer project, Mr. Komkit Lomloy, who is the main contributor in the lung cancer project, and Ms. Piyanut, also in the breast cancer project, and other colleagues. I also would like to acknowledge funding sources from my university, King Mongkut University of Technology, Thonburi, NSTDA, and Thailand Science Research and Innovation Basic Research Fund. And finally, I would like to thank again the organizing committee for inviting me to give a talk today, and also everyone for your participation. Thank you very much.
Thank you for your time. I introduce myself. My name is Ibrahim. I come from Atlanta University from Health Medical Science. Let me ask to Prof. Maman about network pharmacology because I'm very interested about network pharmacology research. I have three questions, and one is, how can we predict a specific target gain for additives in the visualization of cytoscape? Because when we see that the discrete visualization display on cytoscape, display all target gains obtained from all bioactive components in general from a compound, Can it also be used to see more specific compound target related to disease correlation? And second, how do we obtain a compound to then search for its target gain? And third is, can we use target component from different planes to then use a literature review to find target genes. Example, I have one plant because but is very different variety maybe. Okay, that's all for me. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, please. Repeat again.
Oke, let me repeat my ask. Maybe Indonesia Permaman. Oke, pertanyaan pertama. Mohon maaf langsung berubah ke bahasa Indonesia. Pertanyaan pertama, ketika kita melihat network farmakologi di visualisasinya, itu kan visualisasinya secara general. Jadi kayak menampilkan komponen bioaktif dari general komponen bioaktif yang ditemukan begitu ya. Apakah bisa ketika kita menggunakan ketika kita menggunakan visualisasi network farmakologi di Citoscape itu secara spesifik langsung ke disease-nya kita gitu. Karena sebenarnya kalau misalkan visualisasi itu hanya di komponen bioaktif secara general, step itu maksudnya seperti bersifat opsional. Itu yang pertama. Yang kedua, bagaimana cara kita buat mengcollectingnya, buat mengumpulkan komponen bioaktif dari suatu plant, dari suatu tanaman, apakah bisa dari literatur review atau dari GCMS atau ada hal-hal yang lain yang kedua. Nah yang ketiga, apakah dari etik penelitian diperbolehkan menggunakan misal komponen bioaktif kita mengumpulkan dari literatur review tanaman beda varietas, misalkan tanaman ini dari varietas luar negeri, sedangkan kita akan meneliti untuk varietas di Indonesia. Apakah dalam etik penelitian network farmakologi itu diperbolehkan? Terima kasih.
Thank you. Okay, please, Dr. Dina, can answer the question.
Okay, it's very interesting question about the implementation of the networking pharmacology to look about the screening activity in multi-component ingredient extract. Yes, I think the networking pharmacology is one tool to look about the possibility the ingredient in the extract is targeting in the gene, and then also to look upon the targeting in the transcription of the gene, I think the protein. Yes, this is I think very complicated, yeah, because many other concept in networking pharmacology. We look in my PowerPoint. One possibility is the compound is interaction with the one target, and possibility also one compound is the interaction in more than two target, three target, and so on. I think it's the complicated visualization about the networking pharmacology, but I think the networking pharmacology also the clustering is the many analysis pathway possibility, because we have to the clustering. For example, oxoberberin is come from the Archaea flava. Is when we are analysis, we can look upon the possibility inhibition. For example, there is the inhibition about the anti-apoptotic mechanism. For example, BCL2. Yes, this is a clustering. The one mechanism is very interesting. And the second mechanism also in oxoberberin is also inhibit about the, for example, the protein, influence in the angiogenesis. For example, is VEGF, vascular endothelial growth factor. Yes, I think the strategy how to look about the networking pharmacology is very complicated, but I think it's we can the clustering the final analysis, for example, is I think is better. But I purpose for you, I have the suggest for you about when we have the networking pharmacology, I think not the stopping the resistance, but I think we are going to the look about the possibility in vitro activity and then we in vivo activity also. But I think the networking pharmacology is one of the screening dry lab, for example. Yes, and then we also tandem by the molecule docking also, with to look about the many other protein, many other molecule target. And then the last question, what possibility when we are going to review article come from the many other research come from the other country, and then to look about the possibility, the networking pharmacology, I think this is the better also, because we are going to look about the many other mechanism, yes, many other ingredient come from the many other plant come from the foraging research. I think the better also to look how to the study about the implementation of the networking pharmacology pathway, because this is one also is interesting, molecule docking, pharmacology networking, and then also we are going to look molecule dynamic also is, I think, the better to look together. Yes, molecule dynamic to look about the RMSD, and then also to look about the RMFD fluctuation, how to interaction protein with the metabolic one is have the stable in the many other condition possibility. I think better I look the book come from the lecture come from the IK is also is release about the interaction of molecular dynamic. Yeah, possibility is very interesting, because my PhD student is have finished also look isolation ingredient compound active from the Actorus calobus, to look about the in vitro activity about the inhibitory nitrogen oxide, and then we also to look about the molecule dynamic possibility. I think it's better to look how to the possibility, the mechanism of the ingredient come from the compound active in the extract, traditional medicine in Indonesia. Thank you for your question. Yeah.
Thank you for Sukardimat for the detailed answer. Okay, next question from the online audience. In the chat box, there is already one audience member asking. I would like to ask to Mr. Tirapan. My name is Anna, an undergraduate pharmacy student from UIN Malang. I have one question regarding your presentation. What do you see as the biggest challenge in translating this computational model into real-world clinical application? Okay, please, Dr. Tirapan can answer the participant question.
Yes, all right. Thank you for the question. It's a very interesting question, and it's actually the main pain point of this field of research. So yeah, you are right that when we study in vitro data like cancer cells under the laboratory environment, we can control most of the unrelated factors, right? But when we try to translate what we predicted to clinical application, there will be a lot of factors like personal, genetic, or non-genetic profiles of individuals, or the lifestyle or the environment of each patient. So the trend of research right now is that we try to incorporate as many as factors of the individual subjects or patients. For example, we can combine genomics data, transcriptomics data with the clinical data from each patient, like the X-ray pictures or the test from the lab. And hopefully if we can combine this multimodal data and use mathematical modeling or AI or machine learning to identify certain patterns in this big data that we collect from the patients, then we can propose a more personalized or better treatment for individual patients or subjects. So that's a very big trend. There's a trend called digital twins right now that we try to translate each individual patient into a computer model, right? So we represent individual patients with a model, very customized model, and then we can use that model to try to predict the outcome of different treatments, different lifestyle that patient would have, right? And then see which treatments and with the combination of other interventions or the adjustment of the lifestyle would be the best for individual patients. Yes, thank you.
Thank you, Dr. Tirapan, for the answer. Next, that concludes our first session from Prof. Sukardiman. Focus on natural product driven drug discovery for NCDs. The key methodology is network pharmacology and integrated in silico screening, followed by in vitro and in vivo validation to discover active compound from medicinal plant such as Arcangelicia flava and determine their molecular mechanism. It concludes from Dr. Tirapan emphasize the application of computational modeling, AI, and network analysis to unravel complex gene or protein regulatory networks in cancer. The goal is to identify novel therapeutic targets and effective drug combination for triple-negative breast cancer by analyzing signaling pathway and their crosstalk. Overall conclusion, both presentation speakers for the use of sophisticated technologies for network pharmacology and in silico modeling to understand disease complexity at the molecular level and accelerate the discovery of more effective drug candidates or treatment strategies. Thank you once again to our speakers, Prof. Sukardiman and Dr. Tirapan. For Prof. Sukardiman, you can step down from the next agenda. We can invite the committee to present the certificate appreciation to our speaker. Okay, this is all from me. Thank you for Dr. Tiravan, and thank you participant, and I truly apologize for any mistake during this session. Wassalamualaikum.
Thank you very much.
Thank you so much to Mrs. Belia. Ladies and gentlemen, we will now begin our coffee break. Please help yourself to take available food and drink. The sponsor presentation from Albab also starts soon. Thank you.
Next, we will proceed to a presentation from one of the parties who have supported our event this morning. To Wiralab, you may now take the floor.
Baik, terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Saatnya iklan lewat. Karena tadi kan sudah mendengarkan paparan yang sangat menarik dari Prof. Sukardiman, kemudian dari Prof. Tiravan juga. Nah, dari penelitian beliau pastinya menggunakan juga peralatan-peralatan laboratorium yang sangat canggih tadi seperti yang disampaikan oleh Prof. Sukardiman terkait LC-MS. Nah, di sini kami mencoba memaparkan dari PT Wiralab salah satu distributor untuk alat termosaintifik yang kebetulan memang support untuk di Fakultas Farmasi ataupun di penelitian riset lainnya. Baik, Wiralab sendiri berkantor pusat di Jakarta, tapi kami ada brand office di Surabaya. Next. Nah, ini visi misi kami selain distributor juga kita membuka jasa training juga untuk kedepannya jika membutuhkan training dan juga untuk instalasi dan lain-lainnya. Next. Nah, ini beberapa instrumen kromatografi yang biasanya banyak digunakan di Fakultas Farmasi. Salah satunya adalah UHPLC, High Liquid Chromatography. Nah, ini sudah ada di lab riset di UIN Malang sendiri. Kemudian kita juga ada kromatografi gas. Ini biasanya untuk nanti kalau herbal, bahan alam. Kemudian kita juga ada IC. Ini biasanya untuk uji bromat. Kalau Bapak Ibu nanti kedepannya ada pengujian terkait lingkungan, saat ini sih kebanyakan di air mineral, itu untuk pengujian kadar bromatnya. Next. Nah, kemudian yang tidak kalah penting adalah trace elemental analyzer. Jadi untuk pengujian logam berat, biasanya di obat itu impuritasnya, kemudian logam beratnya juga akan diuji. Kebetulan biasanya menggunakan alat AAS ataupun ICP-MS. Nah, kemudian kita juga ada untuk instrumen life science, mass spectrometry. Jadi ini adalah tingkatan di atasnya dari HPLC sendiri, yaitu LC-MS system yang tadi oleh Prof. Sukardiman kebetulan digunakan untuk penelitian beliau. Nah, ini kita bisa mengetahui jejak ataupun marker dari si sampel tersebut menggunakan LC-MS system. Next. Nah, berikutnya kita juga ada spektrofotometer. Ini sangat basic sekali pastinya untuk farmasi dan pasti rekan-rekan semua juga sudah menggunakannya. Kemudian kita ada organic elemental analyzer. Ini biasanya untuk pengujian protein total ataupun CHNSO, sulfur, kemudian protein totalnya hanya N nitrogennya berapa, itu bisa menggunakan organic elemental analyzer atau flash smart. Dan kita juga ada untuk water analyzer seperti pH, itu biasa standar. Kemudian ada turbidity, kemudian ada juga autotitator. Mungkin adik-adik semua titrasinya masih menggunakan manual. Nah, kita juga sudah ada yang sistem otomatis biasanya untuk QC. Next. Nah, ini selain dari kelengkapan unit, kita juga supply untuk kebutuhan consumable-nya. Pasti ada yang namanya vial, penggunaan, kemudian reagennya. Nah, kita juga support untuk kebutuhan sehari-harinya. Next. Nah, ini juga ada untuk apa? Reagen. Biasanya kan ada HCl, H4. Nah, kita juga supply untuk kebutuhan consumable untuk reagennya. Ini untuk preparasi, karena untuk diingat teman-teman semua, bahwa penelitian itu tergantung dari proses preparasi sampel. 70% data dari penelitian adalah tergantung dari bagaimana kita mempreparasi sampel. Secanggih apapun alat seperti LC-HRMS, High Resolution Mass Spectrometry, yang harganya mungkin 19 miliar, tapi kalau proses preparasi yang dengan hanya alat misalkan 20 juta, hasil itu tidak akan akurat. Jadi semua dari proses hasil penelitian itu, instrumen itu hanya memegang peranan sekitar 20%. 10% adalah human, dan sisanya adalah bagaimana kita mempreparasi si sampel tersebut. Jadi ini sangat penting. Jadi teman-teman semua nanti mungkin kalau ada penelitian, pastikan preparasinya sudah sesuai sebelum kita injek ke unit alatnya. Oke, next. Nah, ini pusat layanan kami. Kita ada training dan demo center juga yang ada di Jakarta. Kemudian biasanya memang ada beberapa customer itu selalu sebelum membeli alat pasti mencoba, kemudian menguji si sampel tersebut sebelum mereka membeli alat. Next. Oke, next. Oke. Nah, ini divisi bisnis kami lain yaitu Potamia. Ini biasanya digunakan untuk uji kualitas air lingkungan, yang memang banyak kan penggunanya adalah di dinas lingkungan hidup, karena mereka dipasangnya langsung di site seperti di sungai, kemudian di sistem pengolahan limbah untuk di pabrik ataupun di swasta. Nah, ini salah satu pengembangan unit bisnis kami yang saat ini memang lagi tren, yaitu kita punya laboratorium research jasa dengan menggunakan teknologi LC-HRMS terkini, yaitu Orbitrap 240. Jadi ini adalah lab riset kami yang ada di Jogja. Jadi teman-teman jika ada penelitian mengenai metabolomik, proteomik, lipidomik, nah itu biasanya memang saat ini belum semua laboratorium di universitas sudah mempunyai alat yang untuk HRMS ini. Nah, itu bisa mengirimkan sampel ke salah satu divisi kami, yaitu di Corporate Science. Memang ini lebih ke arah jasa untuk research center. Jadi nanti penelitian teman-teman jika tidak ada alat yang berkaitan tadi dengan lipidomik, proteomik, nah itu bisa juga terkait dengan halal. Nah, karena juga kemarin kita juga kerjasama dengan UGM itu untuk mengenai halal center. Next. Oke, intens. Ini adalah biasanya kita ada sesi kayak Zoom, pelatihan via Zoom. Biasanya kita lakukan itu di hari Sabtu. Memang topiknya akan selalu berubah-ubah sesuai dengan perkembangan saat ini. Kayak seperti kemarin mengenai mikroplastik, ada cemaran mikroplastik di air dan lain-lain. Nah, itu kita akan selalu update setiap minggu, kadang setiap bulan untuk perkembangan informasi yang terkait ada di lingkungan sekitar kita. Baik, ini kontak kami. Jadi nanti mungkin teman-teman jika ada ingin diskusi terkait peralatan ataupun yang lainnya, bisa juga menemui saya, ada di depan. Jika ingin konsultasi tadi terkait penelitian juga, nanti bisa juga konsultasi kami dengan di depan. Baik, terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Thank you so much to Wira Lab for the presentation. Distinguished guests, we would like to express our deepest gratitude to all parties who have supported and contributed to the success of our event today. We extend our appreciation to Nura Gemilang and Wira Lab for taking part in making today's event a success.
Ladies and gentlemen, we now proceed the second plenary session of PLANAR 2025. This session will be discussed two main topics. The first topic is chemical and formulation innovation in translation medicine, from molecular design to drug delivery for chronic disease, and our second topic is pharmacogenetic and AI in translation medicine, toward personalized therapy for systemic lupus erythematosus. This session will be led by our remarkable moderator. Before we begin, please allow me to briefly read the CV of our honorable moderator, Apotheker Alma Nuril Alia, Magister Farmasi. She is a lecturer at the Pharmacy Department, Faculty of Medicine and Health Sciences, Maliki Islamic University. She completed her Bachelor of Pharmacy and Apothecary profession at University Gadjah Mada, and later earned her master degrees in Pharmaceutical Science from University Erlangga.
Her professional experience includes serving as a production staff at PT Surya Prima Natuna Sidoarjo and as a research assistant at the Cancer Chemo Prevention Research Center, Gadjah Mada University. She has been actively involved in numerous research projects focusing on natural product-based therapeutic strategies, and she has produced several scientific publications in reputable journals, including studies related to cancer therapy and neuropathic pain. We respectfully invite our moderator, Apotheker Alma Nuril Alia, Magister Farmasi, to begin the second plenary session. To Ms. Alma, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Okay, good morning everyone. Welcome to the second session of fifth PLANAR 2025, and thank you for the opportunity. Let me introduce myself. My name is Alma Nuril Alia. I will be your moderator in this session, and in this session we will have two speakers. The first speakers coming from Associate Professor Dr. Solejing. We will join us virtually, and the second speakers coming from Dr. Apotheker Yen Ari Indra Wijaya is here with us today, and we are very happy to have both of them with us today. Before we begin, let me briefly introduce our first speaker. She is Associate Professor Dr. Solejing, is an Associate Professor at University Kuala Lumpur or UNIKL, Malaysia. She holds a bachelor degree in Chemical Sciences, a master degree in Pharmaceutical Chemistry, and a PhD in Pharmacology Sciences. And her expertise include natural product chemistry, pharmaceutical chemistry, and pharmacology, and she has an extensive record of teaching, research, and editorial work, and more than 30 scientific publication and in the field in chemical chemistry and pharmacology in areas such as anti-inflammatory agent, and then anti-cancer, and then antioxidant, natural product, and drug formulation. She also served on multiple scientific committees, accreditation panels, and has received several national and international awards. And her talk today is the title Chemical and Formulation Innovation
in translation medicine from molecular design to drug delivery for chronic disease. We would like to welcome our speaker to deliver the presentation, and the presentation time is approximately 30 minutes. Since Dr. Seo is joining us via online, let us make sure the connection is clear. Hello, Dr. Seo. Is the sound or connection is clear?
Okay. So can you hear me? Morning. Can you hear me?
Okay. Hello, Dr. Seo.
Hi, good morning. Can you hear me?
I'm sorry, I cannot hear the sound. Maybe the IT team can help first. Okay. Hello.
Hello, can you hear me?
Can you check the sound, please?
Yeah, I can hear you, but can you hear me? Testing, testing.
Okay.
Can you hear me?
Is the sound clear? In the Zoom meeting, maybe?
Hello, testing. Can you hear me? Testing, testing. Can you hear my voice? Okay, thank you.
Okay. For Dr. Seo, the presentation time approximately around 30 minutes. And for our honorable speaker, Dr. Seo, the time is yours. Thank you.
Okay, thank you. Okay, good morning, everyone. So thank you for being here. So I'm Xiao from Faculty of Pharmacy and Health Sciences, University Kuala Lumpur, Royal College of Medicine, Perak, Malaysia. So thank you for the opportunity from PLANAR 5 to let me talk here today. So I will share the general insights on how chemical and formulation innovation in translation medicine, but it's drawing from my perspective as natural product chemist, just like the Prof. Sukadiman just now. Okay, we are both natural product chemists. Okay, so next slide, please. Okay, so before we begin, so just a quick disclaimer. So this presentation is intended purely for academic and educational purpose. So the views shared today are my own and do not necessarily represent the official positions of my institution or collaboration. Okay, next. Okay. So over 70% of the global death, they are caused by non-clinical. Sorry, hold on. It's very noisy from my side. Yeah. Can you all hear me? There's a noise from my side. Okay, so you can hear me clearly. Okay, I will proceed. So we are talking about the NCDs. So we can see there's over 70% global death are caused by non-communicate. Okay. Please click because I have the animation for the slide.
Check. For the IT team, please next the slide.
Can I take a control of my slide? Okay, can you see it? Okay. So sorry for the delay. So over 70% global death are caused by the non-communicated disease each year. So there's nearly 43 million people annually. So this includes all the non-communicated disease such as cancer, diabetes, and cardiovascular disease. So these current treatments, we face the limitations like poor solubility, low bioavailability, drug resistance, and the reduced patient compliance. So therefore, there's a need to have an innovation both in chemical, design, and formulation to overcome these therapeutic barriers. Okay, so for translation medicines, so it's about what we discover in the laboratory at the bench into real treatment that reach patients at the bedside. So this we call it as a bridge that connects scientific innovation with clinical application. But this process will require multidisciplinary effort from chemists to clinicians. Okay, so as a natural product chemist, so we always think the natural product research, there's a cornerstone of pharmaceutical innovation. So we will say natural product chemistry. Natural product has historically been the most productive source of active compound and leak structure for the drug delivery and drug discovery system. So this slide shows us how natural product drug development has evolved over thousands of years. So from the ancient time, it's based on the observations and practical experience. So all the people, they just collected the herbs, tested them, and identified what worked. Then moving to the 20th century, the approach became bioactivity oriented. So we need to have a systematic extraction. Then we start with the purification and some chemical synthesis. Then we combine with the in vitro and in vivo, which will lead to the clinical trials. So by the 1980s, so the drug development has shifted to target-based drug discovery. So we will screen thousands of compounds just to check on one successful drug. And today, we are in the era of AI. So we were integrating, like just now the speaker from Thailand and Prof mentioned. So we are now integrating genomic, proteomic, metabolomics and the large data sets from our chemistry side. So we will help to predict active compounds faster, so we can reduce the trial and error. So in short, we can see from this slide, so the natural product drug development, they has moved from experience to experiment and to precision, and now to the intelligent AI assistant discovery. So now let's of the role of chemistry in translation success. So our chemistry will start from target identification to synthesis, then move to the chemical optimization, in which we can improve the potency, stability, and the ADME properties. Then to the formulation, so we can ensure the molecule, they can reach the right tissue effectively. So this slide shows to us how natural products move from crude plant extract to specific bioactive compounds like polyphenols, alkaloids, flavonoids, terpenoids, essential oil, and so on. So once we identify the compound, so we will incorporate them into the advanced nano formulations. So after that, this system can enhance the solubility, stability, and targeted delivery. Okay. So here we're sharing some of the iconic natural product success story. So many of our best drugs actually began as natural molecule. So I think this is a well-known anti-cancer agent from Pacific Yuba that everyone knows. Then we also have the artemisinin, which is the Nobel Prize-winning anti-malaria compound. And we move to the ribastine, and now it still remains as one of the most powerful chemotherapeutic agents. And today, I think many of us who are doing nano formulations, curcumin from turmeric is very well known in the nanotechnology and nano formulation. So this example shows why natural product chemistry remains important to drug discovery. But with all this success, our natural product research and development still face some challenge. So the isolation and characterizations can be labor intensive. We face a difficulty to isolate the high yield of the compounds. Then our original, the traditional formulations, they always have the poor absorption, rapid metabolites, and systemic side effects. Okay. And all of this, then we also have a problem how we standardize it. So the questions become, how do we translate promising molecule into the real therapy for our real application? So that we have a strong need to create an innovative chemistry and formulation-driven solution. So fortunately now we have all the advanced in chemistry and formulation that can help us break these barriers. So let's move to the chemical synthesis, the foundation of the optimization of the drug molecule. So for chemical synthesis, we will first start to ensure or enable the precise molecular design. This will help to improve the functions, which we have a systematically tailored molecule, so we can help to optimize the effect. Okay, next on the product innovations. So the product strategy, they can enhance the absorptions and reduce toxicity. And next, on the sustainable method, we want to make sure that the method is consistent and environmentally responsible production. So this comes to the idea for the green synthesis. Okay, so talking about the chemical, the structure, the green synthesis. So we start with the chemical modification strategy first. So first we have the semi-synthetic derivatives. So they can retain the core natural activity while improving the pharmacokinetic performance. Next on the product design. So they can solve the solubility and permeability challenge through strategy masking. So lastly is on the conjugation technique. So they will enable the targeted delivery. Okay. So once we confirm the chemical, we confirm the structure, we modify it. So we will next to move to the green and sustainable chemistry in natural product synthesis. So this is the central part of all the natural product synthesis. So we are now more focused on the eco-friendly synthesis rule. We try to replace all the hazardous reagents with the atom efficiency, the waste minimizing reactions. And we also focus on the biocatalyzed innovations. So we will introduce the engineered enzyme. They will enable the highly selective transformation under the mild condition that the traditional chemistry cannot achieve. And the solvent-free or aqueous phase reaction. They will help to eliminate the volatile organic solvents, so it reduces the environmental impact and production costs. So with this strategy, they can support to all the scientists, especially in natural product chemistry field, the cleaner, safer, and more sustainable drug development. So let's move to the novel technique for extraction, purification, and characterization of phytochemicals. So for extractions, the common used techniques, we have the supercritical field extractions, SFE. We have ultrasound assisted extraction, UAE. Microwave assisted extraction, MAE. Meanwhile, we also have the enzyme-assisted extractions and the pressurized hot water extraction. But these two is less widely adopted. So SFE, UAE, and MAE is the rapid processing, and because they can preserve the phytochemicals integrity. Okay. Next, we move to the purification and characterization. So I just now share with the sponsor, the Villa Lab. So they have a wide range of the equipment, right? So for purification and characterization in natural product chemistry, our main focus, LCMS and GCMS. So they are still widely used as our analytical platform. But then we also have some recent innovations, including molecularly imprinted polymers and the solid phase micro extractions. So these two will enhance the selectivity and sensitivity. So they will be more efficient downstream profiling. But still LCMS and GCMS will continue to be our standard before we proceed to the other method. Okay. So done with the chemical formulation, chemical point of view. Now we move to the formulation technology for enhanced drug delivery. So why formulations matters? Okay. So the control release, they can improve the therapeutic windows and reduce dosing frequency. Next, the stability enhancements, they will extend the product shelf life and protect the active compound. For sure, the formulations can enhance the bioavailability, so they ensure the adequate absorption and therapeutic impact. So as we see, improving solubility and stability is very important, and nanotechnology has revolutionized how we achieve that. So here we have some example of the nano carriers. So all of this allow us to engineer drug system. At the molecular scale, we can offering the remarkable control over delivery, release, and targeting. So we start with the lipid-based nanocarriers. So this system, they form the backbone of many modern deliveries innovation, especially the limbosid. Okay, so it can encapture both hydrophilic and lymphophilic drug, reduce the toxicity, and supported the targeted therapy, especially in cancer and chronic disease. We also have the solid lipid nanoparticles, which offer a good stability and control release profile. Next, on the nano emulsions, this can enhance the solubility and absorption, particularly of poorly soluble drug. Okay, next we will look at the polymeric nanoparticles. So these polymeric nanoparticles, they offer customized and precise drug delivery system. So we will start with the biodegradable polymer. So we will have the safe and biocompatible carriers that can support our sustained release. So we're making them ideal for long-term management of the chronic disease. Next, on the similarly responsive polymer, this allows the trigger drug release at the right place at right time. Last but not least, about the conjugated polymers. So this enhances the drug loading and targeting delivery. Okay, next we will explore how the formulations once even further enable the target and control drug delivery. So beyond we just simply improving the solubility, so the next frontier in formulation is precision, right? Precise, we need to make sure it's precise. So our formulation is delivering at the right dose, at the right site, for the right duration. So this is why the control and targeting delivery system become transformative. So we will target the specific site, so to allow the drugs to accumulate preferentially at disease tissue. So we minimize the exposure to our healthy organs. Next, on the responsive carrier. So they will offer on-demand release triggered by pH or enzyme. Next is on the sustained release. So this system will maintain therapeutic levels over extended period. They reduce the dosing frequency and improve the patient compliance. So this approach is essential for chronic disease that require long-term treatment stability. So by combining the targeting and control release, we can maximize the therapeutic efficacy and minimize systemic toxicity. This is our main target to balance in chronic disease management. Okay, so far we have discussed how formulation strategy refine the delivery and performance. So next we will see this innovation translate to the real-world clinical success story. So I'm sharing with some of the examples. So many phytochemicals like resveratrol and berberine, they are pharmacologically potent, but they are suffer from the poor water solubility, instability, and low bioavailability. So for this very well-known polyphenol of its cardioprotective and anti-cancer property, it has very low oral bioavailability due to the rapid metabolism, but with the nano formulation technologies, so we can increase its bioavailability up to 15-fold in clinical study. So all of this is supported by the review paper, so you can have a read on this. So this improvement clearly shows how formulation can unlock a compound's full therapeutic potential. Okay, similarly to the berberine. So this is a natural alkaloid with anti-diabetic and antimicrobial effect. So the traditional berberine, they have less than 5% oral bioavailability. Okay, but the nano emulsion and polymeric nanoparticle formulation, they boost the bioavailability to nearly 38%. They also demonstrate 7.6 times higher absorptions and better metabolic stability. So this is a report scientific study that support it. So this makes a berberine a strong candidate for translation development once formulation barriers are overcome. I think this applies to some others compound as well. Okay. So we are talking about chronic disease today. So I will touch also about the innovations in cancer, diabetes, and cardiovascular. So for cancer, we are targeted nano carrier system. For diabetic, we are checked on the sustained release and smart response formulations. For cardiovascular, we also develop an advanced nano and lipid-based carrier. So let's have a look. Okay, so in cancer treatments, so they have a scientific report about it. So the nano carrier, they are playing a major role in improving how chemotherapy drugs are delivered. So by packaging this agent into nanoparticles, we can concentrate the drug specifically at the tumor site while we minimize the exposure to healthy tissues. So this not only enhances the effectiveness of the therapy, but also significantly reduce the toxicity and side effects. So you can see overall the nano carrier-based system, they are reshaping the cancer management by making treatment safer and more precise. So all of it is the latest publications from other researchers. Okay. Let's now turn to insulin delivery. So diabetic therapy benefit greatly from advanced formulation system. So smart insulin technology, they will enable the real-time dosing and improve the glycemia control. So this innovation can enhance the patient adherence and reduce long-term complications. So you can see all of this is the latest review paper on the smart insulin system. Okay, Next, move to the oral insulin formulations. So we're protecting the insulin from enzymatic degradation in our GIT system. So they will allow the peptide to be absorbed effectively. So they will improve the comfort and compliance for the patient. So here is some of the paper to support the statement. So the oral delivery, they can mimic the body natural insulin pathway. They're supporting more physiologically and stable glucose regulation. Okay, next in insulin delivery for wound healing. In wound healing for diabetic patients, innovative insulin delivery are making a significant difference. So you can see they have developed of the microneedles in diabetic wound care, how they engineer the nanoparticle therapy for impaired wound healing, and some of the recent advanced formulations. So we have all of these, the localized and control release insulin formulation that can be applied directly to the chronic wounds. They help regulate inflammation, promote cell growth, and speed up the tissue repair. Okay, next move to the cardiovascular disease. So in cardiovascular care, the drug-coated stent and controlled release cardiovascular formulation improve the treatment outcome. So these technologies, they deliver the therapeutic agents directly to the affected vessels and maintain a sustainable drug presence over time and improve the long-term recovery. So this shows the major advancement in chronic cardiovascular therapy. So all the chemical and formulation strategy we have discussed, whether nanocarriers, smart delivery system, or control release technologies have been proven to increase drug bioavailability up to 40%. So we can see this improvement is very crucial because the higher bioavailability, they will translate directly into stronger therapeutic outcome in chronic disease management. So this will show to all of us how thoughtful the chemical design and innovative formulations that can transform a promising compound into a truly effective treatment. Okay. So done with the shared general insight about the chemical and formulation innovation. Here I'm sharing some of my ongoing project at the chemistry and formulation as well. For chemistry focus, my team, we are working on designing new molecular derivative targeting cancer and inflammatory pathway. So we are developing some of the derivative and even the curcumin analogs. So we try to engineer it to improve the potency, the selectivity, and the overall safety. So the aim actually is to create more effective lead compounds that can be translated into the targeted therapies for specific disease condition. Okay, next on the formulation focus. So I'm doing also the conjugations to improve the amylin drug potential. And there are 3 short-term grants I received last year. So we are developing also nano-imogel as an antibacterial agent and also focus on the cognitive enhancing delivery system. So we have combined also exercise and the administration of algawood tea in Alzheimer's disease. Okay. So all these projects highlights how we integrate natural products with advanced formulations technology to improve stability, targeting, and therapeutic performance. Okay, so I think almost done. But other than that, we are talking about the chemistry, formulation, even all the research projects. But there's one thing we shouldn't forget, it's about the regulatory and manufacturing perspective. So the successful translation is not rely on science only. We require a strong regulatory framework and disciplined manufacturing practice. So we need to check also the QC, the quality control. We must adhere with follow the GMP compliance. And through the stability study, we need to ensure our product and reproducibility, safety and reproducibility, okay? So that our aims is not only run the test, but we also develop the product, then we can towards the successful to the commercialization. Okay. So looking ahead for the future trend in innovation, the targeted therapy, they will play a bigger role because we need to deliver the drugs directly to the disease cell with far greater precision, improve the efficacy and safety. At the same time, we are focused on the smart material. So the system that respond to pH, temperature, or some biochemical signals to release the drug, only when and where they are needed. And last but not least is about the personalized medicine. So this will be a future of highly personalized and effective treatment strategies. This is also our future direction in medicine. So we need to allow us to tailor drug formulations to each patient's unique genetic and physiological profile as shared by the speaker from Thailand just now. So this is our aim or maybe our goal towards the future direction in medicine. And we also integrating some of the advanced diagnostic and predictive modeling, just like sharing with the previous, like shared by the previous speakers. So we can design the therapies that are not only more effective, but also safer and more as responsive to the individual need. So they were moving us closer to truly precise and patient-centered treatment. So not to forget about the role of AI. So AI is increasingly shaping how we discover and develop new medicine. So I won't go into detail here as this will be covered by the next keynote speaker. But it's clear that the AI-driven innovation will continue to transform our pharmaceutical science as well. Last but not least, none of this will happen in alone. So when we are talking about the translation of medicines, it depends on the strong collaborations across all of us. Okay, from academic, industry, and clinical partners. For sure, the regulatory agency should play the role into this. Okay, so as we come to the end of this talk, I would like to highlight my key takeaway. So chemistry drives molecular innovations. They're opening a new therapeutic possibility. Formulation then transforms this discovery into clinically meaningful therapies. And lastly, collaborations help bridge the gap between our research and the real-world healthcare needs. So together, these elements form the core pillars of translation medicine. So as we close, I want to express my sincere thanks to the organizer for this opportunity. So I warmly welcome collaboration and discussion, whether in research partnership, student supervision, or joint project. Together, we can continue advancing chemical and formulation innovation and build meaningful solutions for chronic disease. Thank you once again for your attention, and I look forward to connecting with you after the session. Okay, thank you all.
thank you so much to Dr. Seo, and please give applause to the Dr. Seo, everyone. Okay, thank you Dr. Seo for the insightful and very well presentation and sharing to us about your ongoing project or ongoing research. And we kindly ask you to stay with us on Zoom for a little while and proceed the second speaker's presentation before moving to the Q&A session. Okay, now we move to our second speaker who already joining with us via offline, and she is Dr. Apoteker Yen Ari Indra Wijaya. She is a lecturer, Pharmacy Department, Faculty of Medicine and Health Sciences, with Maulana Malik Ibrahim Malang. She held a master degree in clinical pharmacy at Universitas Airlangga and complete her doctoral degree at Institut Teknologi Bandung with research focus on lupus nephritis. Her research activity covered around pharmacogenetic, lupus therapy, polymorphism, and clinical pharmacy with publication on antiviral compounds, genetic markers in lupus nephritis, and therapy optimization. She is also active in community service, authorship of pharmaceutical textbook, and health education initiative. And her presentation today is titled Pharmacogenomics and AI in Translation Medicine Toward Personalized Therapy for Systemic Lupus Erythematosus. For our honorable speakers, for Dr. Apoteker Yen, the time is yours. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, all. Alhamdulillah, peace be upon of all. Distinguished chair of PLANAR of this event, honorable speakers, then the attendees, the academic, researcher, and pharmacy practitioners. It is for me to present the research finding in the 5th PLANAR Seminar and Conference 2025. My presentation today focus on pharmacogenetics and AI in translation medicine towards personalized therapy for systemic lupus erythematosus or SLE. Okay, maybe the IT team can share the slide, please. Yeah, the rapid progression of pharmaceutical and biomedical sciences is centered in translational medicine and personalized therapy. This approach demands the integration of molecular research and bioinformatics, artificial intelligence, and pharmacogenetics. Yeah, next slide. Now, in the background, so we can see that the SLE or systematic lupus erythematosus is a rare disease. We can see that the global average of the incidence is 1 in 2,500. And maybe if we see the budgeting cost for the NHS or the Dinas Kesehatan atau Kementerian Kesehatan di Amerika, nah at the NHS it said that 145 million dollar that need to coverage the vaccine and the drugs for the SLE. In Indonesia, in 2015, there are 1,000 in patient with SLE, but in
the next year in 2016 in 2000, yeah, the incline trend for global prevalence of SLE is significant. Next slide. For lupus nephritis, one of the prolongation or complication of SLE that the amount of the severity in class 3 to 4. The class 3 and 4 of lupus nephritis needs the standard therapy that first line is glucocorticoid plus maintenance that IV cyclophosphamide. Next slide. Because the various of standard therapy, we see that they are in the global race and ethnicity. They are Asian, African, Caucasian. That the diagram make sure that various response of cyclophosphamide is different. So we can analyze from the previous research that the 6 months, 2 years, and 7 years survival rate of the renal survival in the lupus nephritis can be differ based on the race or ethnicity. The highest survival is in Caucasian. The second in ASEAN, like Indonesia, and the third is in Africa. So this is the problem of challenge why the response of cyclophosphamide is different. Next slide. So from the background, we can make the target of our research, and the aims of this study is because the various response of cyclophosphamide in the lupus nephritis patient, we can predict what the factor that patient give or bring that next year or next therapy can be improved by the personalized medicine. Next slide. So we conduct a case control study that included 100 and this is the patient of lupus nephritis in Hasan Sadikin Registry Bandung. So in the West Java, there are 1,000 and more patients with SLE. With the inclusion criteria and exclusion criteria, we can recruit 100 patients, and we can evaluation what the effectivity and the adverse effect of the cyclophosphamide, so we can see what the difference from the response and non-response of the case and control study. Next slide. To identification what the markers or biomarkers that we need to predict the next response of cyclophosphamide, we can identify by the data like bank data or SNP, single nucleotide polymorphism in NCBI. If we in class, we can open the source like NCBI. In biotechnology class, we can see that there are a SNP column that make sure every SNP in the gene can affect the expression of the gene. Now, after that, we make sure by the calculate minor allele frequency or MAF and major allele frequency, so we can see the diagram about distribution of the polymorphism in Indonesia. In the measure of distribution, we can see that the major wild type in the East Indonesia or Papua or Irian. Next slide. After we identify what the SNP that we have to use for the specific gene, and we recruit each patient, so we can isolate the DNA and sequencing to know what the sequence of the ATGC in the gene of GSA1. And we use 3 genes, and after that we combine it with the other confounding, like other factors bring by this patient, and we can analyze in statistical what the effect of the effectivity of cyclophosphamide and adverse effect. Next slide. Before we use the frequency of each single nucleotide polymorphism, we have to analyze and calculate the Hardy-Weinberg equilibrium if the genotype, or we can see the wild type polymorphism of the heterozygote and homozygote of genotype. We can see the frequency and compare it with the chi-square of the Hardy-Weinberg equilibrium in the global frequency. So we can conclude that if the data is fulfill the Hardy-Weinberg equilibrium, and see that there are no mutation in the population. So we can use the data for the next analysis. Next slide. And then we analyze the linkage disequilibrium. If we see the data from the one region, we can correlate an association about one region to the other region. We use the linkage disequilibrium, and it is strong if the LD score is more than 0.8 in the population, like in West Java or in Bali. Next slide. After we have the data, the accurate data, we can make the data is possible to analyze in the tools with SNPstat and linked disequilibrium hub tool that make the several SNP is possible to analyze by the region in Indonesia or West Java. So we make the code data in Indonesia. There are 2 tools like this. Next slide. Now we back to the patient characteristics. So the clinical manifestation of this patient, we can make the comparison about the high disease activity and low disease activity. The effectivity of the treatment of lupus nephritis is evaluated by 2. Their disease activity, we use M/2K score and proteinuria. And the other is the evaluation of adverse effect. There are alopecia, leukopenia, gastrointestinal disorder, and amenorrhea. This is for women only. The ethnic people that we recruit is the Sundanese in the major sample of this research. Next slide.
For one gene, like GSTA1, we analyze the effect of polymorphism of GSTA1 in effectivity of cyclophosphamide. First, we determine the position of the SNP that makes the promoter activity is increased or decreased by the position of this SNP. There are 5 SNP of the GSTA1. There are minus 52, minus 67, minus 513, minus 567, and minus 631. After that, this is the free variable or independent variable. Then we make the target is the disease activity and proteinuria. We also combine with the other factors like age, then the protocol of cyclophosphamide and the comorbid like hypertension. Next slide. So we visualization the result of this multiple gene analysis using logistic binary and two-way ANOVA. So we can conclude that the polymorphism of GSTA1 in the position of minus 513 Ag was associated with reduced cyclophosphamide effectiveness with the odds ratio is 0.19. So the effectiveness is lower 5 times than the wild type. Then the GT genotype at minus 631 is increased the progression of lupus nephritis by the anemia with odds ratio is 2.41 times comparison with the TT genotype. Next slide. Next slide. After that, we use the in silico so we can answer why this polymorphism affect the effectiveness or the progression of the disease by predict what happened to the change of the nucleotide base in the gene that makes the transcription of promoter region is different. So the change of the A to G in the position of minus 513 increase the GST1 promoter activity. So the metabolism is decreased and decrease the effectiveness of cyclophosphamide. It is also result in the minus 631 in GST1 that increase the activity of promoter and reduce the cyclophosphamide effectiveness. Next slide. For the next project is we use the several SNP in human genome that we can get the frame by frame of the gene that we translational protein medicine by the polygenic risk score or PRS. The PRS is based on the human genome sequence that did to compare with the other population, like an ASEAN compared to Europe and compared to the Caucasian. So we have used the big data for the from the next generation sequencing, so we can analyze with several algorithms of AI or deep learning by the logistic regression, naive bias, random forest, but the best accuracy in logistic regression. Next slide. If we use the AI or artificial intelligence, we can see that there are not only one of the method that use an AI. We have to use an AI with the other method, like clinical and experimental testing and population testing first, then we can use the AI, or AI first, then clinical or experimental or population testing. So this an AI is trending in several journals, and this is top 10 journals that include AI in this research. Next slide. For the autoimmune disorder or autoimmune disease, there are algorithms that ever used in the clinical set in the US, that they use the deep learning model like the convolutionary neural network, CNN, in the multiple sclerosis disease. And we have to know what the program or language of the programming, so we can use the algorithm of each model in ML. So there are regression algorithm, bias, and so on. Next slide. This is the scope of the process of analysis. If we are in healthcare 4.0, we have set first the data, load the data in cloud or in the safety data bank. We have to processing with other language, so the data can set to analyze with several languages for the algorithm in the ML. And the final, we can predict what the result if we choose the target of every prediction of or every result that we have to analyze by get the input data to the end server. Next slide. So I will continue our next project with the PRS. If we correlate the SNP to the other SNP that we have in our body, we have 13,000 of SNP in our body, so we can predict polygenic risk score that can calculate in individual genetic predisposition and a disease. So with summing the effect, we can calculate what the how many odds ratio that we can accepting every treatment or medicine that we use. Next slide. So we need the genomic-wide association study, and the clinical utility of PRS is, the first is risk stratification, and second is early detection and prevention. Like if the participant right now is collect the DNA, and the DNA is testing, we can predict what happen later or the future, what the disease or what the treatment that we can use for the specific disease. And the third is cancer risk assessment, because the cancer is by burden in the cost of healthcare. And the fourth is the psychiatric disorders. Next slide. And we use the PRS because the performance is covers the population of each study. So we need the population first, with the patient with disease or the health or normal human, and then predictive accuracy like ROC, and then the prognosis value, so we can predict the disease or comorbid by score of PRS. Next slide. If this participant interest to try, yeah, please visit this GitHub, so we can learn about the language program or the ML and use the data and make sure that our analysis is comparable with the PRS in other populations. There are two PRS in PLING or precise. Next slide. 4 steps for PRS procedure is the first quality control of the data, then quality control of the target or dependent variable that we use. Then the third is the calculation of PRS. They will result in scoring. And the fourth is visualizing. We use our programs like, because there are different language of the Python with the R program. Next slide. The snip that we use have the code, and each snip has the frequency of MIF or minor allele frequency, so we can insert the data or use the data in a global and calculate the PRS with the summary statistic in specific population first. Then, next slide. We have to reduce or remove the mismatched snip. Mismatching snips is like the slipping or flipping the strand that makes the data is incorrect, like G to C, A to T that ever flip. So we have to check to the sequencing. Next slide. And checklist if there are the duplicate of the snip. So the result is only representative for the one snip or each genome in the human. So we have the comment of the GitHub language to use before it quality control based data is finished. Next slide. This is the ambiguous snip that there are several snips that we use, maybe 5 or 10, so we can reduce it first by selected ambiguous snips and remove it first. Next slide. This is the challenge that if we use the AI and deep learning or ML, that there are ethical and social implications. It brings representative of each population. If the ethical in each country is different, we can't use the data all, and we have to sequencing for the specific population first and get the ethics in a global. And the second is selection bias, because if we use the AI, we can see that there are mismatch, ambiguous SNP, and the other confounding that can come in unpredictable. So we have to finish quality control data first. And then the third is the majority of the PRS study until now focus in the Europe population, and maybe in the future we can use the ASEAN data for the PRS for each disease. Next slide. Thank you very much for this time. Of course, our research or our project is not only me. There are many supervisors that helped me and teach me about this polymorphism in pharmacogenetic. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much for Dr. Yan Yan for your informative and very insightful presentation. And now we will open the Q&A session for both speakers. For anyone who would like to ask the question, please raise your hand and mention name and your affiliation before asking question. And for the online participant, you may also type your question in the chat box. And for your information, selected questioners will receive a little reward from the committee. So, is there any question maybe from the offline participant? Okay, thank you.
Thank you for the opportunity. My name is Syahrin Azril Izul Haq, pharmacy student from class 2022. My question for Apotheker Yan Yan. Why does the response to cyclophosphamide treatment for lupus nephritis vary so much across races? And what biological factor likely underlie this difference? Thank you.
Great question. Thank you for this question. The answer is why the response of cyclophosphamide is different. Yeah. In the other models, we can that the protocol of cyclophosphamide, there are two types. National Institute held cyclophosphamide that used in 500 milligram per kilogram in each patient. That the second is Euler protocol that used in 500 milligram in each patient. So if we compare the doses, they are different. But if the patient use these two types of these doses and the effect is different, we can see that there are minor difference. But the second model is there are the side effect that unpredictable come from the cyclophosphamide, like alopecia, amenorrhea, GI disorder, and leukopenia. If the dosage is high, or we can see there are increase of doses in first, second, and third treatment of cyclophosphamide, so can predict that the factor is in doses. But in this sample of this research, the dosage is stable and the effect or adverse effect it not approached. So the remission is the target of the cyclophosphamide. It can be approached by its patient. So we predict it by the factor in each patient, like the genetic. So the predisposition of cyclophosphamide non-response or the decrease of effectiveness is the first, the choosing of the protocol, then the dosage of each patient, and the genetic polymorphism. Thank you.
Okay, thank you. Is that clear for the questions? Okay, thank you. I think there is a question from the online participants for Dr. So. Is Dr. So is still joining us in Zoom?
Yes, I'm here still.
Yeah, the questions. Wait. The question is coming from Alif Bina. It says, thank you, Associate Professor Dr. Seo, for your engaging presentation on formulation innovation. We have several questions to bridge the gap between laboratory discovery and real clinical practice, especially regarding implementation in Indonesia. The first question is, in the context of hard-to-cure chronic disease like cancer or diabetes, can you share a case study illustrating how patient quality of life significantly changed after receiving nanocarrier-based therapies developed in this research, such as resveratrol or berberine nanoparticle? And then the second question is, considering the 19-fold increase in resveratrol bioavailability and the 7.6-fold increase for berberine, what is the percentage increase in efficacy that directly translate into significant long-term clinical outcome improvement beyond just enhanced the drug kinetics? And the third question is, the key goal of nanocarrier is to minimize systemic side effect. However, are there any long-term safety profile that needs to be addressed before large-scale clinical adoption, especially in vulnerable patients with cardiovascular disease? Thank you. For Dr. Seo, you can answer the question. Thank you.
Okay. Thanks for the questions. Can you hear me? Okay, so I'll answer for the first questions regarding the patient quality of life. I think there's a currently lack of published human case study or specific clinical trial result focus on this. So in the research in this area, we are still focused on the preclinical or still in the early stage of human trial. So we more focus on the proof of concept, safety, and pharmacokinetic rather than the long-term patient quality of life. Okay, so maybe I think in future with all the large data set, maybe we can have developed the QOL. Then the second questions about the percentage. Okay, I think it's not possible to provide a specific percentage in increasing the long-term clinical outcome, because I think this is subject to the ongoing research, right? It's very specific and dependence on the specific clinical application and individual patient factors. So we can only check on the enhanced kinetics, provide the potential for the improved efficacy for the certain case. Okay, but again, as I mentioned, with the help of all the AI and the ML, the machine learning tools, in I think maybe in 5 to 10 years, we have a chance to develop for that long-term clinical outcome too. And again, for the third question, it's about safety profile. Okay. So for the narrow carrier, I think this is our main concern for formulations. It's about the long-term toxicity study. So we are still need to conduct the animal study. So to understand the full safety profile of our formulation before we proceed to the real human trial. And for sure, the most important one is about the biocompatible design. We need to make sure the material with high compatibility and we can predict the degradation profile. So we are still, I think this is the ongoing strategy of all the scientists. We are still trying to perform a research to minimize the adverse reaction. And last but not least, I think it's about the characterization of the narrow carrier and the interaction of it with our complex biological system. So I think that's all from me. Thank you.
Okay, thank you for Dr. Sel for your answers. And for the next agenda, we kindly invite the committees to come forward, maybe to present the certification of appreciation to our speakers. Maybe we can start with Dr. Seo first. I need help for the IT team to share the certificate first. Okay. And for the certification is as shown on the screen. Let us take the documentation photo first by 3, 2, 1. Okay, thank you for Dr. Seo. Okay, and for Apothecary Yen, we kindly invite Prof. Iha to step forward and give the certificate to Dr. Apothecary Yen. Thank you. We would like to express our sincere appreciation to Associate Professor Dr. Seo Ying and Dr. Apothecary Yen for their valuable and informative presentation. Okay, thank you for Apothecary Yen. And before we close this session, let me briefly highlight the key points shared by our speakers today. From Dr. Seo, we learned how innovative chemical design and advanced formulation technologies such as nanocarriers, products, and targeted delivery system can overcome key limitations in conventional therapies and significantly improve treatment for chronic disease such as cancer, diabetes, and cardiovascular disease. And from Dr. Apothecary Yen, we learn and gain insight about how genetic variations influence treatment response in lupus nephritis and how tools like pharmacogenetics and AI-driven polygenic risk score can guide more precise and personalized therapy. And we hope that this insight have given all of us a clearer view of how scientific and technological information continue to shape the future of personalized medicine or therapy. Okay, thank you again for both of speakers and for the participant who joining this session, and that's all from me. And I truly apologize for any mistake that I make in this session. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
We respectfully deliver our thanks to our remarkable speaker and moderator. Distinguished guests, we would like to express our deepest gratitude to all parties who have supported and contributed to the success of our event today. We extend our appreciation to Gemilang and Wiralab for taking part in making today's event a success.
Ladies and gentlemen, we will now conclude the plenary session with a closing prayer. The prayer will be led by Mr. Muhammad Amiruddin Licentiate. To Mr. Amiruddin, the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Astaghfirullahaladzim, astaghfirullahaladzim, astaghfirullahaladzim li wa li jami'il muslimin wal mu'minat wal muslimin wal muslimat al ahya'i minhum wal amwat. Allahumma ya rabbana lakal hamdu kama yanbaghi li jalali wajhika wa 'azimi sultanika. Allahumma fasalli wa sallim ala nabiyil karim sayyidina wa habibina wa syafi'ina wa qurrati a'yunina rahmatil lil alamin sayyidina Muhammad ibn Abdullah wa ala alihi wa sahbihi ajma'in. Allahumma ya hayyu ya qayyum bi rahmatika nastaghith a'inna ala dawami dzikrika wa syukrika wa husni ibadatika. Allahumma ya karim ya rabbal alamin, Engkau yang memudahkan setiap jalan menuju kebaikan, termasuk jalan ilmu yang menolong banyak manusia. Ya Rabbal Huda, yassir lana fanna tarkib ad-dawa'i wa anzimata tawsilihi waj'al nataijana sababan lisyifa'i warrahmah li'ibadika as-salihin. Ya Allah, Tuhan pemberi petunjuk, mudahkanlah bagi kami seni permulaan, obat dan sistem penghantarannya, dan jadikanlah hasil penelitian kami sebagai sebab penyembuhan dan rahmat bagi hamba-hambamu, ya Rabbal Alamin. Laksana molekul alami yang Engkau simpan di dalam dedaunan, ranting dan akar, tiada satu pun yang tersusun tanpa hikmah dan kadar. Ya Allah, Engkau yang menanamkan bioaktif pada setiap ciptaan, yang menulis potensi terapi pada fitokimia dan metabolit kecil yang tidak kelihatan. Allahumma ya Khaliq, ya Khaliq, ya Khaliqal mawadi wal asrar, علمنا حكمة اكتشاف المركبات الطبيعية وافتح لنا أبواب الإلهام في تحويلها إلى أدوية نافعة شافية واجعلها سببا في علاج الأمراض المعدية وغير المعدية وتخفيف محنة المرضى. Ya Allah, Tuhan kami, Tuhan yang menundukkan ilmu, yang menundukkan dan memberikan petunjuk, permudahlah ilmu bagi kami ya Allah untuk memajukan negeri dan melayani makhluk-Mu. Berkahilah perjalanan farmakologi, translasi hingga penelitian kami, termasuk Sampaikanlah pada tahap penerapan dan manfaat, dan jadikanlah obat dari ciptaanmu sebagai penyembuh hati dan jasad. Allahumma rabbat taskhiri wal huda, sakhir lanal ilma linarfa'a biladana Indonesia wa takhdum al-kaka wa barakillahumma lana fi masaris saydaliyati tarjamiyah hatta tasila buhusuna ila marhalati tatbiqi wan nafa' waj'al dawaa mimma khalaqta shifaan lil qulubi wal abdan. Bagaikan gen yang saling bersapa, membentuk jejaring penuh rahasia, protein pun menari dalam takdir, mengatur hidup dalam harmoni cipta. Ya Allah, Engkaulah arsitek seluruh sistem ini, pengatur setiap syarat dan sinyal yang ada ini, yang menyembuhkan tanpa batas dan memberi hikmah pada ilmu yang kami cerna. Allahumma ya nur as-samawati wal ard, alhim fahman yuniru turukat ilm, tahlana abwab ar-rahmah fi fahmi syabakatil jinat wal proteinat, waj'al ilmana sababan li shifai ahlil amradil mu'diyah wa ghairil mu'diyah. Ya Allah, Tuhan yang punya segala hikmah dan takdir, yang maha berkuasa, integrasikanlah bagi kami iman kepadamu dan ilmu dalam keselarasan yang indah. Berkahilah acara planar kami yang kelima ini, artikel dan penelitian para peserta dan pendidik kami yang ada di sini, hingga kami memahami jejaring regulasi gen protein, dan jadikanlah ia sebab pencegahan dan penyembuhan penyakit menular, terutama yang menular. Dengan izinmu ya Rahmanu ya Rahim. Ya Allah, Engkau yang menulis takdir dalam helix gen, dan Engkau pula yang menanamkan tanda-tanda kebijaksanaan pada setiap variasi genetik yang membentuk respon obat manusia. Dalam era ketika algoritma membaca jejak biologis atau hayat dan kecerdasan buatan menenun pola-pola yang tak terlihat, kami memohon petunjuk-Mu ya Rabb, agar ilmu ini menangkap yang tersirat dan tetap menjadi jalan rahmat bagi mereka agar tetap sehat. Allahumma ya bari al-asrar, alimna fahmat taghayyuratil wirathiyah isti'malaha fil ilaj syakhsi, wa barik fi khutuwatina wa khawarizmiyatina hatta takshifu ilajan yakhifu fihi alam al-mubtalin litamhil mana'ati waltihabiha. Ya Allah, pengungkap rahasia, ajarkanlah kami ya Allah memahami variasi genetik dan menggunakannya untuk terapi yang dipersonalisasi. Berkahilah langkah dan algoritma kami sehingga lahir pengobatan yang meringankan beban para penderita gangguan autoimun dan peradangannya. Ya Rabb, Engkau yang mengizinkan kami membaca bahasa tubuh pada level molekul, mengenali biomarker yang bisu, dan menafsirkan sinyal biologis melalui kecerdasan yang Engkau titipkan. Di antara model prediktif dan simulasi klinik, di antara data yang berjejaring dan algoritma yang menduga, kami sadar bahwa kesembuhan tetap hanya berada dalam genggaman-Mu. Allahumma ya hikmah, yassir lana jami'il bayanat wa tahlilil anmat waj'al al-dzakal istina'i AI alaina sababan li taqribi al-ilaj syakhsi wathquf fi a'malina barakatan tusa'idul marda ala tuma'ninatil qalbi wa shifa'il jasdi. Ya Allah, Tuhan yang punya hikmah, yang maha bijaksana, mudahkanlah kami mengumpulkan data, menganalisa pola dan jadikanlah kecerdasan buatan sarana mendekatkan terapi personal, serta tiupkanlah keberkahan pada upaya kami agar membantu pasien meraih ketenangan dan kesembuhan. Allahumma ja'al jam'ana hadha jam'an marhuma wa tafarruqana min ba'dihi tafarruqan ma'suma la tada'dana dhanban illa ghafartah wa la hamman illa farajtah wa la daynan illa qadaitah wa la maridan illa syafaitahu ya akramal akramin. Rabbana atina fid dunya hasanah wa fil akhirah hasanah wa qina adzaban nar wa adkhilnal jannata ma'al abrar ya aziz ya ghaffar ya rahim wa taqabbal minna bi barakatil Fatihah. Wassalamualaikum warahmatullahi taala wabarakatuh.
Thank you so much to Mr. Amiruddin for the prayer recitation. May the PLANAR 2025 event brings benefits to all of us. Amin ya rabbal alamin.
The next agenda is photo session. We kindly invite all participants and committee members to join the documentation session.
We kindly ask all committee to come forward. Once again, we ask all committee to come forward for the documentation session.
Baik, mohon izin diinformasikan kepada seluruh panitia untuk dapat maju ke depan untuk melakukan sesi presentasi bersama. Terima kasih. Sekali lagi diinformasikan kepada seluruh panitia untuk dapat maju ke depan untuk melaksanakan dokumentasi bersama. Baik, mungkin dengan untuk dokumentasi izin memandu dalam hitungan 3, 2, 1. Sekali lagi, izin memandu dalam hitungan 3, 2, 1. Sekali lagi 1, 2. Baik, izin memandu dalam hitungan 3, 2, 1. Baik, mungkin untuk yang selanjutnya akan dilaksanakan dokumentasi bersama dengan peserta. Mungkin kepada yang terhormat seluruh panitia bisa berputar badan, dan kepada seluruh peserta mungkin bisa berdiri. Disilakan mungkin kepada seluruh peserta bisa mengisi ruang-ruang kosong yang ada di depan. Mungkin kepada seluruh peserta bisa merapat ke tengah supaya tidak terlihat kosong. Terima kasih. Baik, izin memandu. Izin memandu dalam hitungan 1, 2, 3. Sekali lagi dalam hitungan 1, 2, 3. Oke, dengan gaya bebas. Dalam hitungan 1, 2, 3. Sekali lagi. Oh, sudah. Sekali lagi, mohon maaf, dengan izin memandu dalam hitungan 1, 2, 3. Baik, saya ucapkan terima kasih yang sebanyak-banyaknya kepada seluruh peserta dan juga seluruh panitia untuk waktu yang telah diberikan.
Ladies and gentlemen, thank you very much for your active participation and attention throughout the previous session. We have just completed a very insightful discussion and a wonderful photo session together. Now, we will take a short break, which includes rest, prayer, and lunch. This is the perfect time for everyone to refresh, recharge, and prepare for the next session ahead. For our distinguished guests, the transit room has been prepared on the 3rd floor.
And for our online participant, please take this time to rest and have your meal comfortably from your own location. And I would like to inform you that the oral presentation and poster session will be held on 13th. We kindly remind everyone to return on time so we can continue our program as soon as possible. Enjoy your break and we will see you again right after break up. Thank you. Baik, izin menginformasikan untuk yang selanjutnya yakni sesi isoma, yang dimana kebetulan seluruh peserta disilakan untuk makan, sholat, dan juga beristirahat. Yang dimana untuk yang selanjutnya akan dilaksanakan sesi oral poster yang akan dilaksanakan di lantai 3 pada pukul 13:15. Terima kasih.
me staring at 23 unread messages, namely each response will take
Tick, tick, tick. Tick, tick.
YouTube Music Premium, dengarkan lagu bebas iklan di mana saja. Coba satu gratis.
Cek, cek.
Yes.
Congratulations to best.
Ladies and gentlemen, we have now come to one of the awaited moments of today's event, the announcement of the best presenter. Without further delay, let us continue to the announcement.
For the best poster presenter category,
I'm sorry, there is three category of the best for this announcement. The first category is for the best oral poster category, and our second category it's for the best oral presenter online category, and the last category it's for the best oral presenter offline category.
for the best poster presenter category, the winner is
Badira Kamelin. Congratulations to Badira Kamelin for the best oral poster category. And our second category for the best oral presenter online category. And the winner is Apoteker Muhammad Abbas Ar-Rizik, Sarjana Farmasi. Congratulations to Apoteker Muhammad Abbas Ar-Rizik, Sarjana Farmasi for the best oral presenter online category.
for the best oral presenter offline category. The winner is
Puspita Dianiwati dan Intan Kartika Prabandari. Congratulations to Puspita Dianiwati and Intan Kartika Prabandari. And I say congratulations to the all winners for their outstanding achievement.
We now invite the winners to come forward to the stage. Next, we will continue with the certificate presentation for the winners, followed by a group photo session. We are pleased to invite Profesor Dr. Apoteker Royhatul Mutia, Sarjana Farmasi, Magister Kesehatan, to present the certificates and awards to our winners. The documentation will begin in 3. The documentation will begin in 3, 2, 1. Once again. Oh.
The next, I'm sorry.
For the documentation, in 3, 2, 1. Once again, in 3, 2, 1.
Congratulations to the all winners and our censors. Thanks to the all participants for your active participation.
We hope that these achievements will inspire all of us to continue innovating and contributing in our respective fields.
Ladies and gentlemen, this concludes the 5th PLANAR International Pharmacy Ulul Albab Conference and Seminar 2025.
I'm Ashfa Korinatil Imtiaz, and together with my partner,
Revandi Iman Firmansyah, it has been our honor to serve as your master of ceremony throughout today's event. We sincerely thank you for your participants and attention, and we humbly apologize for any shortcomings. Wassalamualaikum warahmatullahi wabarakatuh. Thank you very much and see you in PLANAR 2026.
Recording stopped.